Ever think about like how cells move around in our bodies? They're like these little travelers constantly on the go.
It's amazing, isn't it? All that movement, it's called cell migration, and it's, well, crucial for so many things. Wound healing, our immune system, even, how cancer spreads.
So how do scientists actually study something so tiny? Cells are microscopic.
Oh, they've come up with some really clever techniques. One of the most basic is the wound healing assay.
Wound healing. Sounds kind of dramatic for tiny cells. What is that?
It's pretty simple, actually. You have a layer of cells all spread out, and scientists, they make a gap, like a little scratch. Then they watch how the cells around it move to close it up.
Like a tiny construction crew rebuilding after an earthquake.
Yeah, exactly. And by seeing how fast they fill the gap, they get a sense of, well, how fast they move, how well they work together. It's a good starting point.
Cool. But watching them in a dish?
Yeah.
That can't be the whole story, right? Our bodies are three-dimensional.
Right, it has its limitations. For something more realistic, there are things like microfluidic channel assays.
Okay, now that sounds high-tech. Microfluidic channels.
Just imagine tiny channels etched onto a chip. Scientists can flow fluids, chemicals through them, create different environments, like mimicking the body.
So it's like a tiny obstacle course for the cells.
Exactly. And since it's transparent, they can watch the cells move in real time, see how they respond to signals, squeeze through spaces.
Wow, front row seats to the amazing race of cells. That must be so useful for studying things like how they move in blood vessels, maybe even tumors.
Absolutely. It's a much more controlled and complex way to study their behavior.
This is all so impressive. So we've got the wound healing assay for a basic look, and these channels for more detail. What else is there?
Well, there's the transwell migration assay. This one focuses on how cells move across barriers.
Barriers. Like what?
Think of a tiny chamber with, like, a porous membrane in the middle. Cells go on one side, and on the other side, something they're attracted to, a chemoattractant.
So it's a test to see how badly they want to get to that target.
Yeah, they have to figure out how to get through that membrane. It's like how they might move through different tissues in our bodies, encountering different barriers.
I can see how this would be important for, understanding how cancer spreads.
You're exactly right. It's a key tool for studying how invasive cells are, how they break through to other tissues.
Getting a bit scary now. But it must also be important for normal stuff, right? Like how immune cells reach an infection.
Oh, absolutely. It's about how cells overcome challenges to get where they need to be, whether it's, good or bad.
So we've got cells scratching, racing, breaking through barriers.
Mhm.
What's next in this cellular obstacle course?
Well, the ultimate challenge, that's the 3D invasion assay. This is where it gets really interesting, much more like what's happening in our bodies.
Okay, I'm intrigued.
Uh-huh.
What does this 3D environment look like?
Imagine a tiny jungle gym made of collagen or metrogel. It creates a 3D matrix, kind of like the stuff around cells in our tissues.
So they're like tiny explorers hacking through a dense forest.
Exactly. And they don't just move, they often have to break down parts of that matrix to create a path.
Wow, so they're not just moving through it, they're changing it as they go.
Right. This 3D assay, it really adds complexity, gives us a much better idea of how they behave naturally.
It's amazing how much work goes into understanding these tiny cells. It's like a whole hidden world.
It really is. And the great thing is, each of these techniques, from the simple scratch to these complex 3D environments, gives us a unique view, helps us understand how our bodies work and how diseases develop.
I feel like I've already learned so much.
Yeah.
But there must be even more to explore.
Oh, absolutely. This field is always moving forward with new techniques and discoveries all the time. It's incredibly dynamic. It's pretty amazing that we can actually see these movements in such detail. Like, we've shrunk down to cell size with a tiny camera.
It really is mind-blowing.
Yeah.
But how do you measure something so small? Track its progress.
That's where the ingenuity of these assays comes in. Each one has its own way. Like with the wound healing assay, you just measure how fast that gap closes. Take pictures, at different times.
Like timing a race, seeing which cells are the fastest.
Exactly. And in the transwell, you count how many cells make it across that membrane. Shows you how many were able to conquer that barrier.
A tiny cellular marathon, only the toughest make it. What about the 3D one? That seems way more complex to measure.
It is more intricate. You might measure how deep the cells go into that matrix, or how many move a certain distance. You can even look at the shape of the invading cells. Some move in groups, others go solo. It tells you a lot about how they interact with everything around them.
Wow, so it's not just speed, it's their patterns, their strategies.
Yeah.
Makes you realize how much is going on that we never even notice. But are there, limitations? Can this really capture everything happening inside us?
That's a good point. They are models, simplifications. They're great for the basics, but yeah, a living organism is way more complex.
Like studying one instrument to understand a whole orchestra. You get the idea, but not the full symphony.
Exactly. That's why, well, scientists are always pushing further, developing new models, trying to get closer to the real thing.
So what's on the cutting edge? What's the next big thing?
One area is organ on a chip models. They try to recreate a whole organ, like a lung on a chip.
Whoa, taking those obstacle courses to a whole new level. Tiny versions of us.
That's the idea. It lets you study cells in a more natural setting, with all the different cell types, the environment of that organ.
This is mind-blowing. How does this help us understand diseases better?
Well, it shows how diseases actually develop at the cellular level. For example, cancer spreading, it's not just random, it's a series of steps with specific interactions between the cancer cells and everything around them.
So it's not just a cell breaking free and floating off, there's a plan.
In a way, yeah. And understanding those steps, well, that could help us block them, maybe stop the cancer from spreading in the first place.
That's incredible. So this isn't just, satisfying curiosity. It could lead to new treatments, therapies for some of the biggest challenges we face.
Absolutely. And not just cancer. Think about autoimmune disorders, problems with wound healing. It has implications for so many things.
It sounds like studying this is really opening up a whole new world of medicine.
It is. And the more we learn, the more we can potentially help people.
Okay, I'm officially hooked on this cell migration world, but I'm also realizing this is complicated stuff. What are some of the big questions scientists are still trying to figure out?
Well, one of the biggest is, how do cells decide where to go? We know they respond to signals, but how do they process that information, actually translate it into movement?
Like cracking the code of their internal GPS.
Yeah, and it's a complex code with tons of signals and feedback loops. Plus, you have to remember, there are so many different types of cells in our bodies.
Right, a muscle cell isn't going to move like an immune cell.
Exactly. They all have different ways of moving, interacting, different jobs to do, so researchers have to think about all that.
It's like each cell has its own little travel guide. Makes you realize how complex and diverse we are, even on the inside.
It really does. And it highlights how much coordination, how much communication is happening all the time, even at this tiny level.
Okay, I'm a bit overwhelmed by all this knowledge, but I'm ready for more. What else can you tell me about this amazing world of cell movement?
Well, let's dig a bit deeper into the signaling pathways. That's how cells get their instructions, their marching orders.
Okay, signaling pathways. What kind of signals are we talking about?
It's like they're swimming in a sea of information, all these signals telling them where to go, when to stop, even how to change shape to squeeze through tight spots.
So it's not just chemical messages. They're really feeling their surroundings too.
Oh, absolutely. They have these receptors on their surface, and they can sense all sorts of things, the chemicals around them, how stiff or soft the material is, you name it.
Like they're constantly gathering intel, making decisions on the fly.
Exactly. And these signals, they all work together. It's like a feedback loop to fine-tune everything.
Okay, so what are some of the big players in this communication network? What signals are really important for movement?
Well, one that's been studied a lot is the Rho-GT pace pathway. They're like little switches controlling the cell's cytoskeleton. That's what gives the cell its shape, lets it move.
So they're calling the shots, making the cell dance.
Pretty much. And you have different Rho-GT passes. Some tell the cell to push forward. Others help it contract, like pull itself along.
Wow, so it's a whole coordinated effort. What other pathways are involved?
Another key one is the PI3K Act pathway. This one gets turned on by growth factors. Those are signals that tell the cell to grow, divide.
So this is about encouraging the cell, making sure it has the energy to move.
Yeah, it's about providing the resources, the motivation to keep going.
It's like the cell's personal cheering section.
Something like that. And honestly, there are tons of other pathways, each with its own role. It's incredibly complex.
It really is amazing. So
Yeah.
We've got the Rho-GT passes for shape, PI3K Act for fuel, and all these other signals working together. It's mind-blowing.
It is. It really highlights how elegant these processes are.
But what happens when things go wrong? With these pathways,
Yeah.
that's when we get diseases.
That's exactly right. When these signals get messed up, it can lead to all sorts of problems. Abnormal growth or, like with cancer, cells start spreading uncontrollably.
Like the communication lines got crossed and now everything's out of whack.
Yeah, it's like the cells aren't listening to the rules anymore. They grow too much. They move where they shouldn't.
And that's how cancer spreads.
It's a big part of it, yeah. That uncontrolled movement is what allows it to invade other tissues. Metastasis, it's called.
So understanding these pathways, it's not just about science, it's about fixing those mistakes, right?
Yeah.
Getting those cells back in line.
Exactly. Researchers are working on drugs that can target specific pathways, block the bad ones, boost the good ones.
Like a toolkit for repairing the communication network.
I like that analogy. And that's how we're getting better at treating diseases like cancer, finding more targeted approaches.
This is all so hopeful. It sounds like we're really starting to understand these processes and use that knowledge to help people.
It's a really exciting time to be in this field, that's for sure.
Thank you for listening to Podcast Learn and Fun. We hope you enjoyed the journey of learning. If you like our videos, please subscribe, click the like button, comment, and share. See you next time.